Friedrich Miesser works in Hauptsael's lab, one of the first in Germany to focus on tissue chemistry that were isolating the molecules that made up cells. Miesser was given the task of researching the composition of lymphoid cells, white blood cells. Friedrich Miesser found an acid substance with nitrogen, N, and phosphorus, P, which he called nuclein.
Walter Sutton proposed that chromosomes are where the factors of Mendel located. Walter Sutton and Theodore Boveri state that Mendelian genes have specific site on chromosome so-called loci. Thomas Hunt Morgan established the chromosomal theory of inheritance with research with Drosophila melanogaster, fruit fly. For his work with Drosophila, Morgan was awarded the Nobel Prize in 1933. Transforming principle was developed by Frederick Griffith in Griffith experiment. Frederick Griffith discovered that the DNA of dead bacteria which is killed by heat can pass on to living bacteria.
Mice were injected with a mixture of heat-killed 3S and live-2R, which type 2R do not kill mice at the beginning of test when it doesn't mix with killed 3S. This shows the survived type 3S DNA had been transformed into the type 3R strain in process of transforming principle. Hershey-Chase carried out by Alfred Hershey proved the DNA is the genetic material in gene and inheritance. Bacteriophage, which injects its gene into bacteria to produce its offspring, is used in this experiment. DNA and protein were labeled with radioactive tags to determine gene which injected into bacteria. DNA was transferred into the bacteria. Erwin Chargaff discovered two rules that helped lead to the discovery of the double helix structure of DNA. The number of guanine units equals to the number of cytosine units, and the number of adenine units equals to the number of thymine units. The relative amounts of guanine, cytosine, adenine and thymine bases varies from one species to another. The most important milestone in the discovery of DNA structure comes when the double helix structure is discovered by Rosalind Franklin, James Watson, Francis Crick, and Maurice Wilkins.
X-ray diffraction is used by Rosalind Franklin and Maurice Wilkins to understand the structure of DNA. Photo 51, an X-ray diffraction image is taken in May 1952. The photo 51 and the other data of Rosalind Franklin later used by James Watson and Francis Crick without permission of Rosalind Franklin and revealed the structure of DNA in 1953.
However, Rosalind Franklin does not getting Nobel Prize because passed away due to overexposure to X-ray.
Before the discovery of double helix structure, there is one structure proposed by scientist Linus Pauling in February 1953, whereby this is a wrong theory made by Linus.